before the long weekend, right?
Yeah.
So I'm going to try, I lost a couple minutes there, I'm going to try to finish out this and get through taste and smell today, which I know is, well, what do you mean 60 slides? It goes faster than you think. That's funny because that speed up adaptation. All right. We were leaving off on this the other day. You have adaptation that occurs. I learned this weekend, when we look at smell, we're going to see that adaptation occurs very quickly. I talked about like walking in the bathroom. And I learned something new this weekend, that my smell to certain smells does not adapt very quickly. Because I went to church yesterday morning, I'm sitting there, these people walk in and sit in front of me. And I don't know what somebody had on, but it was so noxious to me. My eyes were watering. It's all I could smell for the whole hour I was sitting there. It never went away. Like I was getting nauseous. I should have just got up and left. But anyway, and I came up with a name for it during that time. It's supposed to diminish after the first few seconds. 50% of it's gone. That just didn't happen. I don't believe it now. I'm going to teach you that, but I don't know. But I gave them a name. I know what they were wearing. I've never heard of it before, but this is the name. Unrelenting acidic pine. That's what they were wearing. And they were wearing a lot of it. So that's a new fragrance, apparently. So believe it or not, rapidly adapting receptors adapt very quickly. Actually, the key point to think about that is to say, well, what type of pain are we having, what it might be. So when you look at things like touch, pressure, smell, they adapt very quickly. A lot of times when something adapts quickly, that's set up to protect you from that saber-toothed tiger or whatever. If it's a slowly adaptive response, the best example is proprioception. Your chemical composition of blood, great. We kind of understand that goes on all the time. But when you look at perception and proprioception, your body's constantly looking at where it is in space. It's a huge system of different receptors that comprise what we call proprioception. The other one's pain. You'd think, well, wait, if we have pain, we want that to go away quickly. If you really think about that, no, we don't. Because we want to know if there's still damage there, we want to know that. So pain's a way of signaling that, hey, there's something really wrong here. And I'll teach you later about why it doesn't go away that fast. Okay, so we're looking at a somatic sensation. And somatic sensations, touch, itch, tickle, anything associated out by the extremities, the skin, that kind of stuff. And we throw in proprioception back in there. It is a type of somatic sensation. Now, if you're wondering why I keep mentioning proprioception, it's because after we study the ear, you're going to believe in your mind for a second that, well, there are receptors for equilibrium and hearing in the ears. And we'll talk about dynamic and static equilibrium, and you're going to start relating that all this proprioception comes from where your head is. But there are proprioceptive receptors everywhere, like around your joints to tell how fast they're moving. And so you go back and you ask people, and you're like, well, if your head falls over, you'll ride it very quickly. If you've ever seen a toddler, right, when you move them, the head just stays there. We can push them over here. And you do that too, you just don't realize it. And so we think of the head, this receptors, proprioception, as like they actually do adapt pretty quickly. But proprioception as a whole is very slow because that's going on right now, whether you sit here moving your head or not. So as we get through it, you'll see that. So those are your somatic sensations, cutaneous sensations. Then you can classify them also as tactile. That's where we fully get touch, pressure, vibration, itch, tickle. And somatic and tactile kind of go together there. The only difference is we look at sensations is, once again, do they have a separate cell in front of there, or what type of sensory receptor do they have? Somatic and tactile sensations in general have just free nerve endings. Okay. Now there are some of them that are encapsulated, but remember, if they're encapsulated, they just take a connective tissue capsule and put it around it to make it more specific or sensitive for that sensation. It's like taking a grape and putting it over the dendrites. That's what it looks like to me in a grape. If we focus just in now and say, well, what about touch, tactile receptors subcutaneous to the skin. Something's touching your skin. Then we drive it down and we look at it and say, well, is this a crude touch or a fine touch? Fine touch is going to give you a lot more information, but the reason for that is crude touch is like having somebody close their eyes and setting a pen in their hand. And your brain will start to formulate what that might be. So that pen is sitting there and you say, well, you can tell that something is touching your skin. Something is there. But you don't have all the information. You can't tell what size is it, what it actually is, right? What's its texture. We need fine touch for that. Fine touch can allow you to have the specifics of the location that's touched, the shape, the size, the texture. And so there's more sensations involved in that. But here, when you go into a physical exam, you're going to look at like 2-point discrimination, sharp, dull, and all these, and whether or not we can distinguish crude touch, fine touch. Fine touch with the pen example would be, I'm holding that in my hand, maybe I'm grasping it now, and I can tell. Like if somebody put something in your hand and just laid it there, that would be crude touch. But if you brought in more receptors, like if I picked that up with my fingers and rolled it around, I could probably tell that's a pen. But just laying there in your hand, it could be a pen, it could be a pencil, who knows. Pressure is touch that's felt over a larger area or stimulates deeper receptors, like down to the tactile disc. So the sensors for pressure tend to be deeper into the lower parts of the dermis. So we push down harder and activate deeper receptors, or we push down, and when you do that, it deforms a larger area. So, all right, vibration. Vibration, they don't understand completely. Right? The signals that they know that are or the sensors that are picking up vibration are called corpuscles of touch, Meissner corpuscles, the same ones that did touch earlier, and they detect low frequency. Trying to use the mouse and flip to the other computer. And then lamellated corpuscles that are Pacinian corpuscles, which detect higher frequencies. Those are both associated with touch. But what they found, if those same receptors are activated very rapidly, that is perceived as vibration. And then itch, we think of, we got a bug on something moving, it scratched. But that could be an inflammatory response. It could be part of the, like an allergy. Anything that might cause chemical release to stimulate those itch receptors feels like itch. There's a lot of different things that cause you to itch. For some people, just thinking about itching makes them itch. Right? You'll see patients come in that have chronic condition. They scratch their face up, their arms up. It's a nervous tick for some, but once you start itching an area, right, it's going to itch more and more. Well, for one, you're inflaming that area, but then you just can't get it off your mind. I don't have that problem, apparently. I can just tell myself to ignore things, I think, because my whole family was griping because I didn't get up at 4 o'clock in the morning and replace the battery in the smoke detector. It's like, things like 12 feet high. I'm not getting up on a ladder at 4 in the morning. And they said they were all sleeping, like, it was just every minute, beep, you know those. Slept. They woke me up and said, no, I didn't hear that. And I said, you going to change it? No, it's in my bedroom. And I looked up and said, no, I'm not. Just ignore it. They're all like, we never went back to sleep, and you were just in there snoring. I'm like, I don't snore for one thing, but I was sleeping. Would you change it? Not like the fire alarm was going off, but just once every minute. Beep. Just a little beep. You would change it. I know, apparently they would change it too. They weren't happy with it. But it was Father's Day that morning, so what are they going to say? Tickle. Tickle is not a great thing to ask about because tickle has that word of thought. I don't like thought. Thought means that they haven't fully figured out where and what's. They think it has to do with prenerve endings, lamellated corpuscles. In other words, once again, sensation of touch. So tickle's thought to arise using those. That's not a fact then to me. So they're thought to arise. we could get into some fun with that and say, well, why can't you tickle yourself? I can tell you the real answer. Instead of joking, be serious, because actually now this becomes funny now that I'm going to say it that way. Your body knows that it's your hand. But y'all didn't think about that enough, because that was funnier than you think. How did this morning go? Was it all right? I don't know. They didn't walk on eggshells or anything? Just one. Why would I walk on eggshells? I didn't give it the test. Well, I didn't look, but I hope it went well. If not, treat every test like it's going to get you, and remember that the last test is over. The next one's coming. That's always been my advice for kids. It's kept me sane at this place. Just focus on the next one. So what? Next pitch. All right. Thermal sensations comprise hot, cold, cool, warm. Thermoreceptors are, once again, they're a type of somatic, so they're using free nerve endings. If you didn't know this, you have cold and thermal receptors in your cornea, in your bladder, your skin, your tongue, everywhere. Right? You ever have cold weather hit your eyes and it stimulates that cranial nerve and causes you to tear up because you have sensors there that can detect that coldness. I left it there's a very generalized bullet at the bottom of that. Each sensation is mediated by different specific receptors. I had more on that before, but I thought I can just say it and then you don't have to think about it forever. Well, there's hot receptors, warm receptors, and then there's cold receptors. You can go further into that if you want and say, well, what range is that coolness or hot detected? There is a range of how hot or how cold we can sense something is before we lose that sensation. And there's studies out there where they've stimulated hot receptors with something cold, and people have perceived that differently, like hot or cold, when it should feel the opposite. I'm not going to get into all that. For now, we can detect hot and cold. And then pain. So everywhere but the brain, cerebrum, everywhere in the body has pain receptors except there. We actually call them nociceptors. They do not adapt very well. They're very, very slowly adapting so that you know, hey, yeah, that knife is still in my side, my back, I should say. Once you damage something, you're going to have tissue irritation. If you stab yourself, you go all the way back into when they're talking about the inflammatory response, because that releases chemicals. Right? You have the initial pain that's felt, but why does it stay there? Right? Or why do you get pain that gets worse and worse if the stimulus is kind of gone? We'll see that there are kinins, prostaglandins, potassium ions, and we're going to talk real quickly about kinins. I hate when I can't write on my screen. I didn't bring a mouse in. I have to go get it. So the kinins induce vasodilation, increase blood vessel permeability. In other words, we have an inflammatory response going on. All of those white blood cells, platelets, whatever needs to get to that area is coming to that area. And so we need to release these kinins that will dilate blood vessels that allow things to flow easier. And then you can get your phagocytes can move into that area to help with whatever damage might be there. But you also start to release prostaglandins, and prostaglandins intensify the effects of where you look at histamine, kinins. Right? So they're released by damaged cells. Actually, both of those are going to get released when you damage cells. But if you look at what kinins do versus prostaglandins, you can separate them and say, well, kinins actually affect the free nerve endings, causing, when they stimulate them, you start to perceive that as the pain or the inflammation there. And then prostaglandins come along, and they cause the kinins to hang out there longer. So you would say kinins is what's stimulating the pain receptors. Prostaglandins are intensifying that effect and prolonging it by causing those chemicals to kind of linger in that area. That's why if you have a stimulus and the pain lingers on, or that's why, like, you take something like Tylenol. Tylenol is looking to block the formation of prostaglandins so that you don't have that prolonged pain, or it decreases. we have fast pain, slow pain. I bet you can already guess that one of them is going to be fast and one of them is going to be slow. If I tell you something, pain has to travel on a neuron, if I say it's fast pain, what type of like covering fiber type of neuron do you think it is?
Myelinated.
Myelinated in type A, which is the fastest. Remember when I said just memorize them as A, B, C? Yeah. We did that back then. So that's going to fit with this. I didn't mention again, we did talk about this the other day, the type of nerve fibers that propagate the impulses. So is it type A, is it type B, and where is that pain coming from? That makes a difference too. So fast pain occurs very rapidly. When we look at propagation, it's going along myelinated A fibers like y'all just said. But here's where you bring it back to physical exam. Because you're going to ask your patient, oh, you have pain? Can you describe that pain? Now, do they describe it as acute, sharp, or a pricking pain, like somebody stuck you with an ice pick? Well, that's considered a fast type of pain. It's not going to be felt in deeper tissue of the body. So like cutting yourself with a knife or somebody cutting you with a knife, it's going to be a type of acute pain, which is fast pain. A needle puncture, whatever it might be. So if we compare that, though, in physical exam and say, well, there's slow pain. Slow pain occurs along unmyelinated C fibers. So that pain starts and then it builds till it becomes noticeable in the body. It may be described by the patient as throbbing pain, excruciating pain. This is because this is coming from deeper in the skin, or actually you could think of it more deeper, like the visceral organs, a toothache, a heartache. But a toothache's a great example, because toothaches start, you ever had it? I think I'm getting a toothache, and then it goes on and on until it just becomes throbbing, excruciating. Now, if you really, before we transition, if you want to have some fun, I'm not going to play the video in here because for the sake of time, but every now and then I do. There's a comedian out there called Brian Regan. You ever heard of him? Very funny guy. If you Google Brian Regan emergency room, he describes going to the emergency room, and I like it because you never think about it as the provider the way the patient views it when you go, can you describe your pain? Of course, he's describing it in funny ways. He's going 2 thumbs up, and then he goes into a whole skit about people yelling in the ER and you got to out-moan them and all this. It's the funniest thing I've ever seen. And then he breaks in, he brings up childbirth versus breaking a femur. It's funny. You need to laugh today. It didn't go so well, or you just need a general laugh. We'll laugh at that. I'm not going to play it for you. Excruciating. At one point he says, we playing like that pyramid game. He's giving it to me. You have to go watch. I'm not as funny as him. Pain by location. You notice there's different ways we're dividing this up. So we said there's fast pain, slow pain, and now we have by location. So if you define it and say, well, there's superficial somatic pain. Okay, and we'll break that down. We start to get confused by all this. But we already know somatic is associated with skin, muscle, tissues, tendon, Golgi organs. So that's going to be a pain that's felt out there. Superficial somatic pain. If it's deep somatic pain, that means it's probably going to be down into, and I misspoke there. Actually, I misspoke when you divide them up this way. Let me correct myself. When you say superficial somatic pain, that would be more like, I picture somebody like scraping their arms, like dragging their nails down. If you're feeling it and the muscles, tendons are deeper, that's a deep somatic pain. You'll see I spoke that wrong a second ago. It's because of the way they divide it up. Visceral pain, though, is going to be down into the organs, like a heart attack. I know some of this repeats itself. This doesn't because we go to referred pain. Referred pain is very interesting. Referred pain, if you look at a chart of it, looks like this. And I'll come back to that. But it's a phenomenon of referred pain. Why does it occur? So when you look at areas that normally have very low sensory input, like certain organs in the body, and you look at certain skin areas, like if your heart, well, we can switch and see. If you're having a heart attack and you have pain at your heart, you're not going to feel that right away. You just don't feel it. You may feel bad initially or whatever, but as that pain starts to rise and those vessels are blocked, you're actually going to start, that pain becomes intensive. If it's intensive enough, you will start to feel it. But even before then, you often start to feel it in a lot of people, particularly in men, in their left arm and shoulder, right? Like you see on the chart. And if you look up there in the red, it's in that area. And that's because the somatic sensations for the skin in that area actually enter the spinal cord at the same level as the heart. So if you were to trace both of those back, and so as that pain starts to rise in the heart, the brain starts to perceive that, well, maybe it's coming from here also. And so these can be used to help form a differential diagnosis. If somebody comes in and says, I have right upper quadrant pain. Well, if we look at the right upper quadrant pain on that picture, we can look at it, well, is it the gallbladder, the liver? Right? I do not want you to memorize this chart. I do not think it's in the study picks. I'm sure it isn't, but I don't want you to memorize this. In physical exam, they're going to bring this up and talk about it a lot because it helps. It's no different than learning like the quadrants, so that you know if you're stuck with somebody got shot or a knife goes through the right lower quadrant, you have an idea, a differential of what you might have damaged. But if you look at this chart, it gives you an idea. If somebody has pain in a certain area, their shoulder's hurting, well, wow, if their right shoulder's hurting, they've related that some people, that could be they're having like liver or gallbladder problems. I'm going to finish that out by saying, if somebody's right shoulder is hurting, do they have liver or gallbladder problems?
No.
Probably not. I mean, it's not something, don't walk around telling people, especially you want to get older people worried. They say, man, I have mid-epigastric pain. Now, you don't know this because a lot of y'all are younger. You have mid-epigastric pain, especially if it radiates to the back. That can be a lot of things, but it can be pancreas. The pancreas, if you didn't know, you don't want to mess with it. You get diagnosed with pancreatic cancer. I had a friend of the family who was diagnosed with pancreatic cancer. It was quick. He wasn't around long. Pancreas, I hate the pancreas. We need to just get rid of that. But what's more likely, like somebody has mid-epigastric pain, I'm going to ask them, how about at night when you're sleeping? Your throat ever burn? You ever wake up reflux? More likely that. Could they have an abdominal Sorry, I'm getting way off into medicine. It's a helpful tool. Don't memorize it for me. I don't know if they make you memorize that thing in physical exam. But you're going to hear, the reason I'm talking about it so much is in physical exam and on the pants, and every question you ever answer, you're going to hear all these. 49-year-old patient comes in with a complaint of severe mid-lower abdominal pain. It's going to start out like that. And as you learn more, you get outside of these first classes and you go into your clinical classes, you're going to start immediately as you're reading that question. You say, 9 out of 10 mid-epigastric pain. I'm already going GERD, pancreas, liver, or bile, right? Gallbladder, all of those pop in. I'm not thinking Alex valgus, stuff. Y'all don't want to do medicine today? Hey, let's talk about interesting. Am I talking too fast? I feel like I am, but y'all keeping up? Just trying to get along. Analgesia means you don't have pain or relief of pain. So this is interesting to me. Right? Analgesic drugs can block the formation of prostaglandins. We know prostaglandins prolong the pain by keeping things around like kinase. And so when you take aspirin, ibuprofen, and I always have to say at this point that Tylenol, acetaminophen, and aspirin are not the same thing. Anybody that didn't know that wants to raise their hand. Because not everybody's medical. And if you're not a parent, I learned, you probably don't understand that. I learned that back in 2011. I was teaching a junior class, and I gave a question on a test in A&P II that said, certain things are absorbed in the stomach. A lot of things aren't absorbed in the stomach, but some things are, like aspirin, alcohol, start absorption in the stomach. And I had on there aspirin and Tylenol. That doesn't occur with Tylenol. And they came up to me after. I mean, many people in that class, like half the class. And they were standing, I don't know, they weren't as big as y'all, but they're all around me, grouped outside one of the doors, and they said, but that question's just not fair, because aspirin and Tylenol are the same thing. I'm like, holy crap, how'd you get this far in life? At that point. But those are things that you learn as an instructor instead of a provider, is the bias. That you would assume people know that. But what I learned, honestly, is if you don't have a kid, and you're maybe, and it's what's previous, you don't have a kid or you're not medical, there's a reasonable idea that you wouldn't know that they're different. But at the time, I was dumbfounded. I was like, y'all are messing with me. Y'all are messing with me. So then I had a 15-minute talk on Reyes disease and all sorts of stuff. Just because I didn't want them giving their kids after. Okay, well, when they have them. If they have them. All right, local anesthetics, so how do they block pain? This goes back to what we just learned. When you look at, I think I already told y'all this, but you can prevent the opening of voltage-gated sodium channels by giving somebody a local anesthetic, Novocaine, lidocaine. So at that point, that nerve can no longer send the action potential. Then you compare that to like morphine or other opiates. They just change the perception of pain in the brain. It's no longer noxious. The pain signals are still coming from that area to the brain, but the brain just doesn't care anymore, basically. I could throw a movie quote out there, and nobody's going to know it. If you do, I'll be impressed. You may go northward, you may go southward. I'm going to stay here and walk in a circle. History of the World Part 2, Mel Brooks. I'm looking for a pack of Trojans. I could go on. That movie's. If you've never watched it, don't do it this semester. And if you've never watched it, there's nothing PC about Mel Brooks. He discriminates across the board. He's a hilarious guy. So you can't watch that stuff anymore, right? I just. Proprioception. Everybody's somebody else's idiot. We might as well make fun of each other. Okay, the difference between that and when you're trying to be ugly or insult somebody. All right, proprioception. Head, limbs, everywhere, just everywhere. Just think of proprioception as everywhere. One of the key things I keep driving home is proprioception adapts very slowly because it's continually providing information to the brain. So I discussed this, and now I'm saying that there are proprioceptors just about everywhere, and they're talking about the degree of muscle contraction, amount of tension on tendons. So these are our muscle spindles, our Tendy-Golgi organs. Right? We also can look at the speed or movement of parts, like when you're running, your knees can figure out how fast they're going. The weight of objects, all of that is incorporated into what we call proprioception. We're going to look, maybe this week, towards the end of the week or next, at the ear and start seeing their hair cells in what's called the vestibular apparatus. Anytime you hear vestibular, your mind should automatically think equilibrium. That's what they do. So in the inner ear, you have some receptors called the vestibular apparatus, and we're going to define that out later, and they can tell where your head is in space. All of that. But the three we're going to talk about today, look real quickly again at muscle spindles, tendon organs, and then joint kinesthetic receptors, which actually is a name for something that's not a receptor at all. Not that you could pick it out. I'll show you what I mean. So we already know what muscle spindles are. Muscle spindles are found within skeletal muscles. They can tell how much stretch is on the muscle. they adapt very slowly. Nobody want to question that? We talked about reflexes. Do reflexes occur very slowly? No. Can a muscle spindle that is quickly stretched respond differently than one that's always working? Yes. So in general, they respond slowly. Why? Because even as I'm up here just walking around, you use your hands. I'm not doing a quick stretch, but your muscle spindles are part of proprioception, and proprioception is constantly monitoring the stretch on that muscle. When you strike it with a reflex hammer, that's kind of abnormal. You put it on a quick stretch. So see the difference I'm making? It can react. If I didn't say that later when you're studying, you would come back to that and say, wait a minute. And what also happens is the brain sets, just like we have muscle tone, we also set the tone or the sensitivity of the muscle spindles. How quickly are they going to respond to stretching? That's why some people have hyperactive, hypoactive reflexes compared to the person next to them, but that's not necessarily abnormal. When you start having absent reflexes, now we know something's abnormal. And if you want to take that further, you can say, well, wait a minute. If somebody has hypoactive reflexes, I need to pay attention to that, right? Well, let's say you're testing their right knee, and they have a slightly, what you consider hypoactive reflex. What's the first thing I would do to make sure if I even need to worry about that? Yeah, test the other side. They got hypoactive, test all the reflexes, because they're probably all going to be hypoactive. Right? Doesn't necessarily mean there's a problem. All right, muscle spindles we find everywhere. What we didn't mention earlier is that if you need to control a muscle, you're going to have an increased amount of muscle spindles in that eye. It gives you much finer control. So a good example is biceps versus an extrinsic muscle of the eye. When you look at how many muscle spindles per muscle cells there are in a bicep, it's less per. Then when you look at the intrinsic muscles of the eye, they're very quick in helping do very precise movements to keep your eyes tracking. So there will be more muscle spindles to muscle fibers in those areas to give you that finer control. You just need more receptors. You have tendon organs. Once again, tendon. Oh, yes.
Is it possible to increase the density of spindle fibers in different muscle groups? Just like as a result of like strength training and stuff, or other
I actually think that's a very, very good question, but I don't know. Because I like to tie things into how y'all think and how I'm thinking, because you can increase myofibrils in muscle cells. Can you increase the amount of muscle spindles that are available there? I just honestly don't know. Great question. That's something worth me looking up. I'll ask Dr. Sutcliffe. Maybe he's had that. Maybe he's looked into that before. Can you increase the amount of receptors available for muscle stretch? Sorry, I like that question a lot. I never thought about that. I'll see if I can find us an answer. Tendon organs we know are protecting the tendon. Muscle uses the tendon to attach to bone. There's excessive tension. The tendon organs will pick that up. They are still slowly adapting, and their tone, I didn't put it on there again, their tone is still set by the brain. And then the last one is called a joint kinesthetic receptor. And a joint kinesthetic receptor is not really a particular type of receptor. What it is, if you look at like the knee, there are receptors in and around the knee. So you would say the knee has a joint kinesthetic receptor or receptors that surround it. In other words, if you microscopically went looking for a joint kinesthetic receptor, it doesn't exist. But the combination of those is what they're talking about, because you can tell the pressure, acceleration, deceleration, tension, all in like one joint. So think of a joint kinesthetic receptor as just surrounding a joint and giving you all the information about that joint. Most of the time you're going to see these associated with what's called a synovial joint All joints have capsules, but some joints have an additional layer in there that releases synovial fluid, so they have a synovial membrane. That's it for the opener there, and we switch to olfaction and gustation. You may not think so, but I have really whittled down these next two into pertinent information for them. Right, so they're not very long blocks or sections. That's why I said we'll probably get through them today. We'll see. I'm just kind of rambling, giving you time to reset. Well, I like to do that when we're switching topics. Nobody else had any problems this weekend? Y'all all have perfect lives. I know I wouldn't want to share either with the whole classroom of people. I get paid to do that. But, it cracks me up how many students like talk about it. You lead as crazy as life. I'm like, y'all do too. You're just not paying attention. You're not paying attention to the idiots around you. There's not. I'm always paying attention to them. And everybody has a right to be an idiot. Okay. Chemical. If we say chemical senses, we mean taste and gustation. And we mean olfaction and smell. So those require a molecule. If you didn't know this, right, when you breathe in, you're breathing in molecules. And it may be a unrelenting acidic pine molecule. And it comes into the, yes. It killed my joke, but go ahead.
It's the same way.
Yeah.
Like my dog has skunk Tuesday at midnight last week, and smell never got better at all. I had to put beans out here in my house.
Did you let him roll over the couch and stuff afterwards?
Yeah, he got it like 12 bags. He still smells like skunk. Took it right in the mouth. Right.
So being that we live out in the middle of nowhere, my dogs get skunked about once a year.
It's so bad.
But you know what you got to do? It's baking soda and tomato paste. You take, look at the internet on it, but you take like tomato paste and you rub it all over. It gets a lot of that out of it. Like I've been able to bring them back in the house the same night.
I woke up with him standing on top of me. It smells so bad. I'm like, what the fuck is happening?
We got it on your bed.
Yeah.
I know a good place you can buy a new mattress. I mean, they're not a big box store, but they sell good mattresses for cheaper. Skunk dog out on my bed, it might be gone.
Thankfully, just a sheet. I watched a sheet.
No, it's a 5-alarm fire every time my dog gets skunked. I appreciate you bringing it up because you're right. It doesn't go away in your house. Just smell it for the next month.
I still smell it.
Yeah. You know who smells it even more though? It's the people who don't live there that visit you. You gotta get some Febreze. No, but tomato paste or something like that is what we use. Works really good. Those shampoos they sell for, we couldn't get them to work. Sorry, everybody's like, why are we talking about it? I asked. So chemical senses. There are molecules coming off a skunk that you can smell. So in your chemical senses, when we look at, there's a difference here. I can't underline here. The molecules interact with receptors, and they're either going to produce, once again, a generator potential or a receptor potential. So that means that olfaction has free nerve endings that are picking up molecules. But taste, gustation, has a taste bud, which sits in front of the first order sensory neuron. So that is a receptor cell potential that is generated. We're going to involve some neurotransmitter there. So smell and taste work very closely together, which we all kind of know. We know if our nose is stopped up, food doesn't taste very good or as good as it did. You have a harder time tasting things. Both of them use molecules that in order to perceive them, they must be dissolved in water. Right? Mucus is mostly water. And then olfaction is more sensitive than taste to the same chemical. So we're just going to look at smell for now, olfaction. And we get these, they love to just change things up and say, we can't just say a molecule. We can't say you can smell a molecule. We need a name for that. So they came up with, well, we'll call it an odorant. Just like later we're going to have a tasting. They bring up new words. That way we got to memorize things. Odorants are chemicals that can stimulate the olfactory hairs. I hope so, because they're called odorants. Right? So any molecule that can stimulate, not all can, any molecule that can stimulate an olfactory hair is an odorant. So that means there are molecules that don't stimulate olfactory hairs. Can you think of one main one that your house could blow your house up? Yeah, natural gas, right? It doesn't have a smell to it, but they add sulfur smell to it. Right. And olfactory receptors respond to the same chemical or odorant by producing a generator potential, because we're going to see their free nerve endings. That's called the olfactory response. And we can pick up very, very small differences. Like if you use an example for this weekend, I'm sitting very close to people, right? And my family's all here, and there's all these people. Well, I'm like, these people are right in front of me. I'm like, it's not going to take me long to tell them. They're right. They're sitting right in front of me. The whole time I'm thinking, I just want to let them know, hey, whatever you're using, stop. Fine. But I asked, after we got out, I asked the people that were sitting next to them, and I asked my own family, I'm like, that wasn't bothering y'all? And they're like, we didn't smell anything. So then I'm driving home going, is it me? No. I'm like, y'all didn't smell that? Well, not only can a slight alteration in the molecular structure of an odorant change its smell greatly, people often also perceive different smells differently. How we perceive them. That's an interesting fact of things. It's actually big money. And the last thing, and we'll take a break, is because when you look at concentration differences, not only the molecular structure, but maybe you dilute it out. If you look at indole, which they say is, I've never dealt with indole, but it's a substance supposedly found in both coal tar and perfumes. So those two don't go together. But what they found, and I don't know how they did this, sufficiently diluted, if you take indole, dilute it out, it starts to smell like jasmine. That's big money if you like that kind of info that we're not going to get further into. But if you look at perfumes, they're constantly making new ones, right? Because everybody not only perceives it a little differently, but they need to find that one that everybody likes. So they're taking all these different things like indole and changing their dilution rate or molecular structure a little bit and trying to see if they can create the perfect perfume or cologne that everybody likes. You know, it all smells good. And then you get into like, what do they call it that makes you like sexually stimulated? Yeah, aphrodisiac smells. That's yeah, pheromones. That's what I was looking for. Is that? Yeah. I'll leave it at that. Do they still have like when I was of club age? I know y'all can't picture that. You go into the bathroom. It was always Polo and Jacquard. They still got those things in clubs, like in the bathroom. You put like a quarter in, it has a spray of white Jacquard. You ever seen that? They got rid of those? Good. Good. Because people didn't understand that Polo, not. My buddies were cheap. We got to take it. My buddies were so cheap, though, they would stop. Back then, they had malls. They don't really do malls now. They would stop by the mall and do the free sample before they went to the club. Y'all take a break.
Used to do that. Oh! Let me stop biting my fingers. No.
Your wife.
I love myself.
If they come back,
That's how we all felt. Like, most tests are like, okay, let's pull out the two guys today, like, bucks. I mean, there's like graphs that we studied, one graph has been recorded, like written out, and it just tells it, oh, she's referring to that graph. It's worded poorly. I didn't even realize they were just weird. The straightforward questions, they were able to get.
Yeah. Well, yeah, I mean, I think Dad, I'll shoot you all this stuff. Like when we'll do another intro slide.
Yeah.
Like bring up how one of those like shirts I can Thank you, appreciate it.
You have fast read of it.
You guys meeting this week, or you just stay the whole time?
I'm literally reading.
Okay.
I went to the gym.
Okay. I wish I could, but I don't want to do it. I mean, something for free.
Because I'm not reading. Summarizing everything. It works for me. I know what that will work for everyone. Doesn't really matter.
No. Unnecessary. But you study like you did, no matter which way the information is.
Yeah. And I have like study as much as I do. I'm already waiting.
Crazy, crazy.
They're not for me.
They were egregious.
I don't believe
No, that's not true. I didn't watch Green. I'm sure my daughter scratches. I was like, sugar heavy. Yeah. Waiting for my crash. About one sugar high. They're good. How was the oil that turned out? You used to be traveling. Do like a classic old day. I'll become friends. I know the problem with those though is like now it's like now you gotta have that pot. Yeah, that pot is scary. They're expensive. They're expensive. Did you get the whole setup of the pot, like the strainer inside the pot and everything? Yeah. That's nice. If it doesn't
boil at your house. No, dude, you
probably a lot. Did you do some sausage in it too? Yeah. That's like a low. It's like a low end beef to kind of balance off the expensive seafood, but it's like so filling.
for the peptide. I want to do it up. I've had one crab oil in my life. Like a certain
You have
poured it out on the table.
you have your own crackers and everything. Yeah.
They're
Yeah. I just like just the whole experience. Like, it's like a whole like different like the requirements for like material like to cook and to consume. You know, you can make do like put together, but if you have all the right stuff, it's like a whole. It's a unique set of kitchen utensils. Kitchen hardware.
Usually go there for July.
Oh yeah. The butcher paper, the newspaper. I had a guy in my we were in Virginia, so we were kind of a stomping ground. Like ran the line. With like old like they would like even on the beer it's like what they
He probably has like a title.
yeah, just like a cannibal as a yeah. He single-handedly made me taste the biggest bomb. The heaviest bomb. It's like a bar with vodka. That's all I really have to do is fucking called biggest bomb. I think it had a coloring. It was a different color, or maybe it was just the vodka.
Alright, we are ready.
I feel like I'm gonna throw up. I can't drink another Smartie.
Try to get this orphan to not come out.
Candy.
I'm not even mad if the computer's not working. I don't know. Maybe they can figure it out. Alright, odorants. Odorants. When we look at, we're jumping right back in. When we look at the epithelial of your nasal area, we call that the olfactory epithelium, and how it's set up. And taste and smell, the epithelial areas look kind of similar. So we have three different kinds of cells in both of them. You have the olfactory receptors, you have what's called sustentacular cells, or what's really easier called supporting cells. And then you have basal stem cells. That is very, very unique, and we'll get to it. So I kept referencing this during cranial nerves, that when you look at the olfactory receptors, they're bipolar neurons. That goes all the way back to our types of neurons. You have multipolar neurons, bipolar neurons, pseudo-unipolar neurons. And we said bipolar is going to be associated with special senses. And this is where we see them first, is the olfactory receptors are actually these bipolar neurons. So that means the olfactory receptors are actually the first order neurons in the olfactory pathway. So we have our, you look at the peach colored kind of things here, each one of these is going to have an axon. This axon is going to come off, and it's going to terminate. When we say terminate, what I actually mean, you can use terminate, but it's going to synapse. Terminate means that neuron ends. And then you have a knob-shaped, that's the axon, you have a knob-shaped dendrite that is down, sorry, it's hard to draw, I'm drawing on the scroll pads, down here. And coming off of that, each of these knobs are going to have cilia. I'm going to draw them larger. You're going to have cilia that come off of here and are projecting into the nasal cavity. Right? And for reference, this, remember, this is our olfactory bulb. This bone here is the curviform plate. And the curviform plate has a bunch of Swiss cheese holes in it that allow these neurons to go through there. That's how they're set up. So the cilia is the first time we're going to start to hear the word again, transduction. Okay, every special sense that we study, I want you to pay attention and know where transduction initially occurs. In other words, I'll ask you things like, what is the site of olfactory transduction? Your answer would be the olfactory hairs. And I'll specify that on every sense as we go through it. Yes?
Wait, sorry, just so you said that it's from the peach color cells, the axons, or the blue cells?
Blue. Did I see? Oh. I was drawing on the peach and appreciate it.
Yeah, just making sure.
The blue. Sorry, I messed that up. But I'm not going to ask you to diagram the nasal epithelium. I need you to know what all of them do. But yeah, correct. That was my bad. So coming off of the blue, that's your knob-shaped dendrite with your olfactory hairs coming out. You got to go on too quick, I guess. The key point, though, is that the key points, these are bipolar neurons, and they're the first-order sensory neuron, because they are considered a neuron, and they have these specialized cilia. I call them specialized because when you look at a molecule that's in your nasal cavity there, it's going to go and be dissolved in mucus or water next to these cilia. The cilia then are going to take that stimulus and change it into an electrical potential in that neuron. So that is the site, though. The ciliary hairs are the site of olfactory transduction. That's where it starts, where we take one stimulus and turn it into something else. Okay. Now that we corrected myself, I almost said ourselves, but y'all went wrong. Corrected. When you look at the peach, when there's supporting cells. Supporting cells are just columnar epithelial cells. Remember back to cell types, all columnar epithelial cells generally have some that have specialized into goblet cells. Goblet cells are mucus cells, and mucus cells are what's going to be releasing mucus into your nasal cavity down there, right, so that we can dissolve those molecules. The supporting cells are also surrounding the neurons, and so they help to preserve the electrical potential that's going on there also. Then the last one is really, really unique. So in between these cells, we're going to have little triangle-looking cells that are called basal stem cells associated with the olfactory neurons. So olfactory neurons only last about a month. We saw that greatly with COVID, right? How many people with COVID, one of the common symptoms with COVID was they lose their sense of smell. And because of that, they couldn't taste as well either. Well, we're learning that that's called flavor. There's a mix between them. They didn't really lose their taste buds so much as they can't smell it anymore. But how many people there are some, I know, but how many people are still said, I had COVID, I couldn't smell. They can smell now because they grow back after about a month, because what's really unique is last block I said neurons do not replace themselves. Neurons do not go through mitosis. The only area in the human body they found that you have neurons that are replaced are the bipolar neurons associated with olfaction. They actually have basal stem cells that make new neurons. A lot of study of people studying this and other animals to figure out why. Because that would be something we really want to know. Right? I'm spinal cord injury. That's what I like to work in when I used to have a real job. But if you could figure out how to make neurons grow, that would be very valuable, right? And initially they'd say, well, we see all these neurons die, they don't get replaced. Now don't confuse that with peripheral repair of neurons, myelinated. These are completely replacing these neurons. If you can figure out how to replicate that, you could just leave school now. It could be the Bill Gates of the medical world. Just leave school and do that for the rest of your life. Then you could mess. Whoa. You ever stop yourself from going too far? I just did. Okay, olfactory. I mean, I really did. I thought to talk while we were just standing outside on the balcony. Olfactory glands. Interspersed with these are olfactory glands, right, which is where we find the actual mucus coming from. Now, they can't just call it a goblet cell. They got to say, well, tell you what, where does that mucus actually come from? And some guy named Bowman said, I figured it out. And so now we have Bowman's glands. So Bowman's glands are olfactory glands that are secreting mucus onto the epithelial surface of the nasal cavity. I said it all that way because we're not looking at something that different. Olfactory physiology. When we move fast through olfaction and taste, but it's not as hard to understand, is olfactory receptors are going to react to odorant molecules. In other words, we have to have a molecule that we're able to perceive, or it wouldn't be called an odorant. So that's the stimulus, and it's going to encounter a receptor. You smell something, the molecule comes into your nose, that molecule goes to the olfactory hairs and acts as a stimulant. That's going to cause in that bipolar neuron what's called a generator potential. Anytime you have free dendrites on a neuron doing the transduction or the conversion of the stimulus, you're going to have a generator potential. It's going to generate a potential in that neuron. I'm going to say this so we have the clarification too. If we had a receptor potential, that's where that separate cell comes in. And we're causing a potential inside of a cell first to cause a release of neurotransmitter. That's why they call it a receptor potential. Right. Here we have generator potentials, and that hopefully will trigger one or more nerve impulses. Olfaction has a very, very low threshold. Right, so we got to define things. The way we think about them is something that has a low threshold means you can smell it easier. You can distinguish that smell. So they add methylmercaptan to natural gas because it's a very unique odor. You don't normally smell that, and it's very easily perceived. So you can distinguish that out of the other smells in the area very quickly. It's recognizable, but it's not usual. It's not a usual smell. That way you know when your cat's trying to kill you. My cat tried to kill me with that stuff. That's all right. I turned on the stove, middle of the night, filled my kitchen with gas. All I could hear was click, click, click. That one I got up for. I cleared the house. Hey, so adaptation of odors occurs very quickly, rapidly. So about the first, you don't have to memorize percentages, just know smell adapts very rapidly. Don't confuse that with the whole conversation I had this morning about my smelling and whatever was in the room then. It adapts very quickly. You lose about 50% of that smell within the first few seconds, and then by about a minute, it is completely adapted to that. The better example is the bathroom. If it didn't adapt very quickly, you wouldn't keep walking in some of these bathrooms around here. Okay, so about a minute, you probably don't smell it very well. The olfactory pathway, we're going to go back through this and say, okay, well, we have bundles of unmyelinated axons that are called olfactory receptors that we find here. There's a lot of these. All of these bipolar neurons, axons that are going through that cruciform plate, collectively is what we call cranial nerve 1. So all of those together are cranial nerve 1, and they're also the first order sensory neuron for olfaction. Those olfactory nerves are going to terminate, or in other words, synapse. That neuron ends and goes to another one. So they're going to terminate within the olfactory bulb. So the olfactory bulb, when you say olfaction, the olfactory bulb is where first order sensory neurons for smell synapse with the second order neurons. That's the second neuron in that order, that pathway going back to the CNS. You can ask that another way and say, well, where are the cell bodies of I'm not going to ask you this, but where are the cell bodies of second order neurons for smell located? You'd say the synaptic bulb. I keep saying that. No, you would not. You would say the olfactory bulb. From there, they're going to go back through what's called the olfactory tract. So this would be the olfactory tract going back. And then they're going to go out to it doesn't matter where it's at, but they're going to make their way directly to the olfactory area. So the primary olfactory area of the cortex is where perceptions of smells occur. I can smell that and I know what it is. It does also go to other areas of the brain. But what's unique, there's already been one unique thing about olfactory is that they have basal stem cells, making neurons, but also olfactory sensations are the only sensations that do not go through the thalamus before going to the cortex. The thalamus is relay center, right, for all sensory, but olfaction doesn't go there. Maybe it explains why it's so fast to pick it up and adapt. And then you have olfactory tracts that actually branch off and go also to the limbic system and the hypothalamus. Remember, the limbic system is responsible for our emotional brain, our emotional or memory evoked responses to odors. You can smell something enough and you create a pathway using the limbic system hypothalamus that you put that into long-term memory, and every time you smell that, you remember something, like the bathrooms around here. I always give a better example is my grandmother lived out in East Texas and was a very good cook, as I remember being a little bitty kid, walking down this big long hallway. Right before you got to this kitchen, there's a house my grandfather built, so it wasn't fancy. It was an old farmhouse. But when you got to the kitchen, you would smell things where I love to learn the appreciation of everything deep fried. You'd smell fried chicken, banana pudding, biscuits and gravy. Y'all getting hungry, aren't you? You'd smell that. As soon as you hit that kitchen, every time I just remember, oh, that smells great. But before I got there, she had a laundry room to the left. And what did everybody back then have in their laundry rooms? We don't use them as much. You mentioned it earlier.
Mothballs.
Mothballs. They keep mothballs in there, keep bugs away and stuff. So there's mothballs, and she had a lot of them apparently. When I would walk by, I'd smell mothballs, and then I'd smell country fried chicken steak or something. To this day, when I smell mothballs, I get hungry.
Jeez.
I'm actually hungry now. Just talk. Anybody else in the mood for chicken fried steak? Yeah. Okay, so those, and you have those smells. So I always say the smell remembers when, just like the song remembers when. We all have memories with songs. Kinds of receptor cells. There are 1,000 different types. Think of it like the alphabet. You're going to take your olfactory receptors, and actually in this case, olfactory receptors can distinguish like multiple different odorants. And a single odorant can be picked up by, in the same regard, many different olfactory receptors. So the differences that we have in smells comes from a combination of these receptors. So it's like using the alphabet and saying, well, we have 26 letters. We can make thousands upon thousands of words. We can do the same thing with smell. So you have these scattered throughout. When we look at taste, we're going to see a lot of our taste buds are located in certain areas of the tongue, but in the olfactory epithelium, they're kind of scattered throughout because they can pick up different things. And then we get to the second bullet there, and it says, hey, I got another pee test or something? Oh, I was just kidding. Seriously?
Yeah.
That should piss you off. I don't know. Hope y'all been drinking water. Here's an interesting thing. Once they go in, I'll tell you what's on the test. Just kidding. All right. So think about this. I gave an example of the mothballs and me getting hungry. If this second bullet didn't occur, that would never happen. We would lose memory of responses to the odors because the olfactory nerve for a given receptor type will always connect to just two spots in the olfactory bulb. So every one of those bipolar neurons connects to two spots in the olfactory bulb, and that never changes. That means that each olfactory neuron that gets replaced goes on to connect to the same spot in the olfactory bulb. But from the olfactory bulb back, those don't go away. Only the I hope y'all are getting my visual. These are olfactory neurons going through the curviform plate. The olfactory bulb and tract, the second order, third order stuff out there, that will always stay the same. And it's been mapped out. It's the same in most people and mice and all this. But these olfactory neurons that are going up to it are being replaced, but they always go back to the same synapse area. If they didn't, you would lose that combination of type pathway. Just another interesting kind of fact. So I just said all that. As new neurons are replaced, the roadmap never changes. It connects to the same spots. Don't ask me hypothetical because I know somebody's thinking it. I heard it in my head. You're like, well, what if they don't? Okay, well, what if they do? Well, they do. As we finish out, I told you, smell goes by very quickly for this. When I get to these, I know we're to the end of what I'm talking about with smell. We're going to define out like hyposmia. Hyposmia is a decreased or reduced ability to smell. As you get older, every disorder in the world you can relate to as you get older. But I put it here because if you didn't know, as you age, not only your taste, but your smell changes. And you have a decreased ability to smell as you get older. Now this may be due to the fact that some of those neurons that were getting replaced are no longer getting replaced, or things get old. Maybe they're not getting fully replaced, and you're losing some smells. If you look at head injuries, Parkinson's disease, I love it. They always feel the need to list smoking. And I guarantee you find me a disorder in the world that smoking is not listed as a cause. Yeah. I'm being funny, but at the same time serious. Smoking causes a lot of problems, but it really does affect your taste and your smell. Right? I don't know about how that relates to like vaping and stuff. If it still does that, you'd have to tell me. I have to Google that, I guess. I don't know. Antihistamines, a lot of things can cause hyposmia. Anosmia means it's a total lack of smell. You can no longer smell. you have a head injury that damages those olfactory nerves, like the olfactory tract, maybe you have tumors in that area that damages those tracts. If your nasal cavity is very inflamed and full of mucus, you're going to affect your smell. You could have a total, you see smoking again there, or you see a physical obstruction. Now, I don't see physical obstructions in adults very much, but I have seen it in a lot of kids. Because kids, for some reason, love to take like mashed potatoes and peas, little matchbox toys and things, and shove them up their nose. I guess that's just a thought you only have when you're young. All right, let's see what this does. Now, they'll stuff peas up there, and they'll come in, and people don't know the peas are up there blocking. And you'll look in there and see things are blocking their nasal passageways. Or they could have a polyp. Some people develop polyps. That's where I might see it in adults, like they're saying, I can't smell, I don't have a cold, I'm not sick, and I can't smell all of a sudden out of this. They could have a nasal polyp on that side. The other side's clear or both. You see nasal polyps and peas in kids a lot. And a lot, meaning more than you do in adults. Yes?
Is the smoking because of, like, the smoke is actually, like, doing, like, burning and damage to the neurons?
Yeah, I think it damages the olfactory receptors themselves. Or blunts them. Now, and maybe that they're just blunting their ability to work. If I can use that word. Blunting, probably not the best word to use. I link both of those in my same mind. But also, when I was going to give you a good example. When you smoke, the microvilli, cilia, and things in your esophageal, and you look at your airways, your trachea, they make them where they don't function very well. So when you stop smoking, now smokers have a smoker's cough because they have postnasal drip and things that get down into their airways, and normally we cough and clear those up. But when you're damaging those microvilli, cilia that are along your airways, when you stop smoking for a while, they'll start hacking up a lung, right? Because they start working again. They're there to move secretions up and out so you can cough them out. I'm sure the same thing with the olfactory epithelium. They're making them where they can't function efficiently. Now that's the worst part for a smoker besides the draw. The cough that comes after you quit.
Sort of like this, patients that have a stroke, and they smell like burnt popcorn and those weird things like that. I've heard of that. I don't know if that's something that's
I've heard of
common. Is that something, is that like an issue with the brain or something with the olfactory nerve tract?
I don't know. That's so somebody has a stroke that's having damage to areas of the brain are in some ways being damaged or activated. The better example for me is like migraines. People who have migraines, severe migraines, a lot of times will have abnormal smells, sights, because things are getting activated in the brain itself, or like a seizure. Seizure causes you to smell things that aren't there. All right, what was your actual starting question?
Well, I think it kind of is a dumb question. Like, if there's a stroke, there's an issue with the brain, right? Lack of blood supply, oxygen, so
Yeah. You're just having abnormal firing. That's the only way. They're not really smelling burnt popcorn. Brain tells you are. All right. Zinc deficiency also causes anosmia. You can lose your smell. If you prescribe the right kind of zinc, zinc is a chemical that's used as a growth factor for the chemical senses. That's why if you looked at, once again, not smell, I bring up COVID a lot. If you look at COVID, one of the things people would tell you to do is take zinc. The zinc's not necessarily going to keep you from getting sick and all that, but it is a growth factor for your nasal epithelium. It also helps other areas of the body, but people would take zinc. We don't normally see people walking around with zinc deficiency all the time. All right, taste. Taste, taste, taste. Gustation. There are receptors for taste buds located on the tongue, the epiglottis, oropharynx areas. When we look at these, we already know, and we'll bring them back up, you already know what areas are being picked up from the cranial nerve, or you'll see them again either way. Taste starts to decline with age. Actually, taste changes all your life, from child to adult too. If you look at taste bud anatomy, in naming the cells that are associated with it, it's similar, but its structure is not, because taste buds are cells. So each taste bud has an oval body, has 3 different types of neuroepithelial cells. So it looks like this picture here. And comprised in there, we have gustatory receptor cells, meaning we're going to use receptor potentials. And they're going to have supporting cells that are surrounding them, and they're going to have basal cells. These are base cells, not basal stem cells. These basal cells is a subtype of stem cell, but they're not creating new neurons. You don't know the difference between creating a neuron versus a type of cell that can replicate. In other words, a subtype of stem cells means it would be a cell that's going back into the cell cycle. But you're not creating the new neuron, because we talked about it earlier. So these are basal cells, so if you take those basal cells, you can make more receptors. Okay, so at the top of this taste bud, you're going to have a pore called the taste pore, and each receptor cell has a microvillus that's going to come out through that. That's called the gustatory hair. So the way it's going to work in this case is the basal cells are going to make supporting cells. Supporting cells will turn into gustatory receptor cells. That's the pathway here. The gustatory receptor cells don't last a month like olfaction. They only last about 10 days, which is good because we often eat that pizza before we take it right out of the oven and you're starving. You burn your taste buds out, but they come back fairly quickly and will be replaced. So we have odorants and now we have tastants. Tastants is a chemical that can stimulate a gustatory receptor cell. Tastants, just like odorants, must be dissolved in water. Anything that cannot be dissolved in water cannot be tasted. So your saliva secretions are mostly water. They'll dissolve that in water, and those molecules will be perceived or transducted by the gustatory hairs. So then the gustatory hairs are the site of olfaction. Losing my mind again. Gustatory hairs are the site of taste, gustation. That's where transduction will occur. So this is different than other senses. We're using that cell, and now we're going to explain it. The result of transduction of a tastant creates a receptor potential. Transduction of an odorant creates a generator potential. Transduction of a tastant creates a receptor potential, which is going to stimulate the exocytosis of a neurotransmitter from that cell or that taste bud. Okay. And because what seems obvious to me is not always obvious to you as you learn it the first time, listen closely so you won't confuse this later. We like to talk in terms of first order, second order, third order neurons. Olfactory neurons were first order neurons in the olfactory pathway. When you look at taste, the receptor cell is the receptor. The first order neuron is what it's going to do, release neurotransmitter to stimulate. So a receptor cell is not the first order neuron for or for gustation. Right? So that's what the bottom statement is saying, is neurotransmitter molecules released from the taste bud, or that receptor cell, will cause an action potential, hopefully, in the first order sensory neuron. Keep hitting the wrong button, it skips 2. So primary taste. Now this map that I'm going to show you of the tongue, some people would say is bogus, and they're defined, everything in medicine. Anatomy 1 doesn't change a whole lot. This is more of a physiology thing, though, because Anatomy 1 structures. But they're finding out that taste buds may be more diffusely located on the tongue than we thought. But we're still to a point where we're in textbook medicine, so the way we're still going to study it the way the tongue is set up for the 5 primary tastes. Now, every culture, as they developed, has had what they call taste, whether they understood the tongue or not. Right? And each taste may have an evolutionary advantage. So we have 5 tastes that the modern world pretty much says, yes, these are the things that we can taste. They all make a combination of these. But if you look back to like the Greeks, they had other words for taste, like harsh, bitter, astringent. They had a lot of different descriptions of taste. But these are ours for now. One of them is my favorite. Read through them and you'll see if you can figure it out. This is the classic way you learn taste. You have your taste buds that pick up bitter towards the posterior part of the tongue. And then on each side you have sour. Sour on each side, salty, and then sweet on the tip. Okay, so we're going to look at what's the evolutionary advantage of this, and I'm going to make fun of one part of it, is sweet foods are associated with things that are supposed to be good for you. I know that's good news to hear, right? So the tip of the tongue is where we find taste buds that pick up sweet taste. That's where they're most sensitive. And what we're really looking at is carbohydrates. So for those of you who don't eat a whole lot of carbohydrates, you have an absolute requirement to eat carbohydrates. You cannot do, I think it's, what's it called years ago, Atkins, where you eat protein instead of you cut out all carbohydrates. That's terrible for you. Now I'm all in, because I've never really been on a diet, you can tell. But well, I've never had a diet or anything. I'm lucky in that aspect. But if I did, that would be the one I did. Because I watched somebody on it once, a family member was doing that. They were eating bacon 24 hours a day. I'm all in. I'm all in. I can eat bacon 24 hours a day. I can eat a back package by myself on a weekend. I don't know. All of y'all who are cringing out there, eat right, work hard. Go back. I left part. What part did I leave out? Eat right, work, exercise. Eat right, work hard, exercise daily. Still bad. I'm not an idiot though, and I don't want my last few years to suck. So I'm not going to completely embody that. Sour taste. Sour tastes are associated with spoiled foods. And many foods become bitter or acidic when they spoil. Milk. Right? I'm the person. If it's outdated on the milk carton, I ain't drinking it. I don't care how it smells. My whole family's going to drink it. Some of y'all are too. Yeah. It smells good to me. I'm like, no, we'll get a new one. But that's just me. I also take immunosuppressant drugs. So, you know. But I've always done that. That's a lot. All I ain't drinking. But sour says it's spoiled. So maybe evolutionary, we may not want to eat that. It's on the back sides of the tongue. Bitter. I love this. Bitter tastes are associated with poisonous foods. So anything in the environment that you might eat, or the external, if it's poisonous, it's supposed to taste bitter. And if it's bitter, that's telling you not to eat it. So it's the back middle of the tongue. Now I laugh at it because if I'm putting something that's poison in my mouth, I'd rather it be at the tip of the tongue so I know not to let it get to the back of my tongue. Because once it gets to the back of my tongue, it's almost to the area where you start involuntarily swallowing stuff. How are you going to get it out of there? But it's like this, I guess it's a safeguard. But if you like to really think deep about these things, it's kind of fun. Take any animal that's out there, like a deer, or any animal that's born, they seem to know what not to eat. Now they do follow the example of, like chickens follow the example of the other chickens and stuff. But how would an animal that has no real conscious thought probably about a lot of things, right? Not sitting there reading books on what not to eat. They would eat something that's bitter or sour, they may not eat that. But that's how it tastes to them. So it's evolutionary. Kind of interesting. I just wish more animals would eat cedar bush, but you know, that's just me. I hate cedar. Salty tastes are associated with sodium chloride. Sodium we need in our diet. Another thing that you cannot cut completely out of your diet is sodium. You don't want your sodium levels off. These are the front sides of the tongue, just to the back side of sweet, which is on the tip. And then there's one that's pretty diffuse across the tongue. When I first started in medicine, I'd never heard of this. They added this one later. And I say it funny because that's how I like to remember it. When I read it the first time from East Texas, I said, oh, umami. So it's umami to me, no matter what. But umami, I found out, is one of my favorite taste buds. Because umami is associated with amino acids. Umami is the taste for glutamate in foods. There's no real translation, but meaty, savory, broth-like. And there was a gentleman, I believe in Japan, I took it off my slides, who discovered that he loved the taste, meaty, savory, broth-like, seaweedy stuff. And he made that and found it into a chemical form. Anybody know what that's called?
MSG.
Yeah, MSG, which has gotten, in my opinion, a bad rap for many years. They've proven a lot of the bad things they thought about MSG wrong. I'm not going to say it's good for you. I'm not going to go there. Probably not. But I will tell you this. My daughter worked at Cane's Chicken. And they pretty much soak that chicken in MSG. That's why it's like crack. Because I've learned that I like to taste MSG. But there are newer, for those of you who study, there are newer studies out there too that talk about MSG may not be what's causing your migraine for so many people. That was one of the things, but I still don't know. I have an opinion that it may not be as good for you as people are trying to come back and say it is. I still like it. I still eat Cane's. Receptor location, diffuse. And there's no conventional location. So umami's, the tongue has reception for umami at different, golly, it's diffuse. I've messed that one up before, you can tell, and I almost did it again, and I still did. When you look at taste buds, yes.
So a while ago, in one of my psych classes in undergrad, did.
And then you lost me right there. You had a psych class. You had a psych degree.
Yeah.
Major in psychology. You didn't speak up the first week. I was looking for you. I'm kidding. Go ahead. I mean, I'm kind of kidding. Go ahead.
But we talked a lot about like the different sections of the tongue associated with taste, and something that was interesting to me, I just want to clarify with you: spice isn't considered a taste, right? Is that more associated with pain?
I've thought that same thing and looked it up, and there are people that say that, yeah, sure, pain is associated with that spice. But I'm from Texas. I mean, I'm with you. I don't fully understand it. I'm from Texas, and I eat like what most people call hot or spicy all the time. But I don't think there's any pain there. I mean, ghost pepper's pain. They're definitely, but no, they have found that certain spices activate nociceptors all the time. So, y'all discuss that in psychology? Because psychology covers everything. Man, I can't leave it without saying it. You know why you went into psychology, right? Figure out what's wrong with yourself. Kidding. I'll get it on everybody. I gotta keep going so we can finish tasting or smell the tasting. I joke a lot about psychology, but the truth of the matter is, when all of y'all get to psychology and you read the DSM, what version are they on? You know?
I think it's 5 still.
5 still? 5 texture vibes. There we go. When you get to reading that, and they're going through it, and they're showing you the disorders real quickly as they go through a million of them in psychology class here, you're going to be amazed at how many you have. I'd be like deep into it, like, oh my gosh, say something that I don't have on this one. We're all crazy. We're all just a different kind of crazy. What is crazy? You're not supposed to say crazy, right? I say that every night when I'm looking at Instagram. That's crazy. Dude, it's stupid. Yeah. But I'm glad he did it, because I got to see it on video. Alright, sorry. If I offended you, let me know later. Don't run me down in the parking lot. My truck's probably bigger. What kind of car you drive?
Sedan.
I don't have that big a truck. I'm just kidding. Okay, Papillate. I wouldn't hit you anyway because it cost me too much to fix it. Insurance ain't going to cover mine. Papillate. Elevations on the tongue. So your taste buds are found on these. There are 3 types of pathway that contain taste buds, and then there's one type of pathway that only contains tactile receptors, which goes back to what he actually just brought up, right? Tactile, these types of things. So you have valate pathway, they got funny names. Fungiform pathway because they look like little mushrooms. Foliate pathway. When I said earlier that taste changes as you go from like a child to an adult, most of the foliate pathway are lost during childhood, which in a rudimentary way of thinking of it may be why, like as a kid, I didn't like tomatoes and onions. Now I love tomatoes and onions. And actually, most of my life as an adult, I didn't really like cucumbers. I didn't hate them, but I didn't really like them. Now I can eat cucumbers all day. I love cucumbers. Now that's never changed for broccoli. Broccoli was bad at 3, it's bad at 50. It's the worst thing you'll ever eat. I don't understand y'all. I don't eat a lot of it. I went to the Korean at lunch, I told you. There's so many vegetables. I didn't eat a single one of them. None of them look good to me. No. I'll try anything once though. Didn't change. Except broccoli. I won't change. Broccoli is never going to taste good to me. I don't care how much. Put cheese on it. Whatever. Okay, so those are your. So the 2 you have as an adult are valate and fungiform. Filiform, I know they all sound the same. Filiform pathway are tactile receptors, and where tactile receptors are on them to determine the texture, right? Not the taste. You don't find taste buds there. So if we look at each different type of taste, it has a different threshold. Remember, the lower the threshold, the easier to detect. So what do we want to have be easy to detect? Poison. So bitter things are picked up, have a lower threshold than the other taste. When you look at sour substances, they're a little bit higher than bitter. Right? The remaining are higher than sour. So just to recap that, you would say bitter has the lowest threshold. Sour would be just higher than that, because both of those we want to pick up quick. We don't want to put those in our body. And then when you look at like salty, sweet, they're higher than those 2. That threshold's higher. So it's perfectly safe to eat all the chocolate cake you want. Just throwing it out there. It's not good for your body, I guess, but you can eat all you want. Taste adaptation. Smell occurred very, very, very rapidly. First few seconds, 50% of that smell is gone in most cases. Then over about a minute, being able to distinguish that smell is pretty much gone. But it takes about 1 to 5 minutes for a taste adaptation. Okay, so it takes a little longer. If you're continuously, if you're eating an apple pie contest, you have to take a break. Because if you don't, it'll all just start to taste the same. You will adapt it to it. Last few parts here, I gotta hurry. Is flavor is taste and smell put together. So most of what we say, this tastes good, is a combination of how it smells and how it tastes on the taste buds. That's called flavor. What's interesting is most of this is driven by what's called retronasal olfaction. So as you are walking around and you're, let's say, like normally during this block or right after this block, y'all start cooking a bunch of things, or in between the first 2 blocks, and I can smell that. It makes you hungry. So my brain can tell I'm breathing that in. But when you look at retronasal olfaction, when you start to chew something in your mouth, they call it volatile chemicals. But those chemicals from that food actually go back up into your nasopharynx and go back up and stimulate through the back your olfactory receptors at the nasal epithelium. And the body can tell. So it's going to determine what are the taste buds picking up and what is the retronasal olfaction telling me for smell for that food. Not the smell that's going in. So you go, well, how does it know the difference? Because it responds to both. It's because the body tracks as you breathe in and out. When you breathe out, you push volatile chemicals that you're chewing up into your nasopharynx. When you breathe in, they come through this way. So Pavlov smells. Flavor is developed when you put it in your mouth and eat it. Kind of interesting. Last thing we have to talk about here real quick is the pathway. Taste receptor cells do not have an axon. They're a cell, but they're not a neuron. Only neurons have axons, so they produce a receptor potential that generates the release of neurotransmitters that will synapse with the first order neuron in the gustatory pathway. Okay. So each first order neuron actually synapses with several taste buds. I didn't bring it up on a big picture again because I think we're starting to understand that occurs. Then if you look at the actual pathway, so cranial nerves, we go back to cranial nerves. The facial nerve, cranial nerve 7, does the anterior 2/3 of the tongue. Glossopharyngeal nerve, cranial nerve 9, does the anterior posterior 1/3 of the tongue. And then you looked at vagus, which did the throat and epiglottis. Now we didn't put sensation in there, we didn't put tactile, because we're really talking in this case about taste. So if you look at the taste buds, they're going to go along that cranial nerve. And I told you I'd show this last week. I told you I'd show this later. They go from the taste buds, all three of those, right? And they're going to go to a nucleus in the medulla oblongata. Some are going to project to the limbic system. And then they're going to go up to the thalamus. The thalamus, where all sensory but olfaction goes through the thalamus. The thalamus will relay those out to the primary gustatory area of the cerebral cortex so that we can have perception of what we're tasting. So that's how they're connected. I always like to show the basic pathways when we go through. So that should be it for gustatory or taste. Right on time. Well, I'm 2 minutes in. If I went through it too fast for anybody, get with me. We'll figure it out. If there's any confusing parts there. Like I said, next we'll get to vision. And vision and an image I like to spend a little more time on, so I wanted to get through these a little bit faster. Because there's some things within image that y'all are going to have questions about. But I'm so good at it by now, I'm going to clear up every question before we leave the room. That's why it's going to take us long. All right, y'all have a good day. Have a good lunch.
Hey.
That's what I was trying to remember. They call that math number. It's more like I don't know how to figure out that math. There's some fees of things. They'll come in and say they can't smell their parents. Like green needles. You're gonna see them for sure. Not only that, they put them in their ears. They put mashed potatoes in their ears and stuff. It's awesome. When's the last time? I can tell. When's the last time you had bacon? What year? I'm not talking like bacon, how good this is bacon, good one flavor. I'm talking crispy, one after the other bacon. Now, I can tell you where you are. To you, friend, when I said,
That's I've watched the videos. You're gonna stay oh yeah. Yeah.
you're gonna say, well,
Big cheese up.
I don't eat bacon. You're gonna eat it. Good stuff just every now and then. Every 3 months, that's
too long. You saw that? That's your assumption. Yeah.
Like, did I make dough and then did I use my pasta maker to roll out? It's frozen. It's frozen, and then I put it in the oven, and then put it in the fridge, and then put it in the microwave. Well, we got people calling it an enchilada. My food's getting attacked over here, so.
Don't feel like doing my work study. Beat. I was scared. I mean, like when you walk into the shop, I guess because I like it. Yeah, like he gets there. He was wondering about the OCS. Like he thought we actually were OCS. Like, oh, like how'd you get into OCS? And stuff. No, it's not an OCS. It's listed in the word. I mean, he probably doesn't also know, but like we're the only ones who wear OCS here. Maybe the actual OCS again. Yeah. Well, I think they all use it.
That's the one that's just
Other people in the diamond?
Yeah.
Was it
Oh no, these are the. This is,
all the milk?
yeah, this is the farm. I thought I was the food guy.
You probably just sent them in.
I don't know what part of A&P, general chemistry, physics, general psychology, stats. He is GRE. Yeah, oh, you're thinking GRE. That's all it counts, right? Like the application manager is split up different. It's just those two different things.
How much did you apply to now?
You just put them up, you don't have to. Notice there's an system. You can only do this to your own. I didn't expect you to come back. Yeah, I did.
This is